Hello, everybody. Today we're going to get into some of the history and science that came out of the work of Gregor Mendel. I'm sure all of us have heard of Gregor Mendel before, but our goal is to sort of understand the science that he did and the contribution to heredity and genetics that science led to. So, Gregor Mendel was around in, I think his work came out in the late 1860s, and so to put that into some kind of historical context, right, you'd be looking at right around the Civil War time. Charles Darwin would have been a contemporary. Abe Lincoln would have been a contemporary. So just to give you some sense of when this was. He worked as a monk in Austria, and he was in charge of the gardens. And he actually liked studying a lot of different things. He was really a naturalist. He had bees. The head of the monastery didn't like the bees, right? He raised mice. But the work that he's most known for are his pea plants. And so I want us to understand the why of that, right? Pea plants are different than other organisms when it comes to being able to study them for genetics. So, why pea plants? They're small, they're easy to grow. You get a lot of offspring. I mean, if you think about every single pea inside of one of these pods over here, right, it could be a new plant. So you get a lot of data to work with. They're relatively easy to grow. You can actually get a couple seasons of them. They have short seasons, right, to get from seed to seed again. So you get quick results. And in many ways, they're a model system. Probably one of the biggest things that's sort of alluded to here is that mating is easy. And that has to do with this, right? We couldn't find this in mice or bees or humans. And that is flowers, angiosperms, plants that have flowers, many of them are considered bisexual. And what does that mean? Well, it means that they have both male and female reproductive cells as part of the same organism. And in fact, some of them, including peas, can actually fertilize themselves sexually. So if you think of it, pollen is sort of the sperm of the plant, and there's essentially a egg or ovaries down here. You can actually take the pollen from one plant in peas, touch it to the stigma here, and you can actually get these seeds to fertilize. Right, so they're sexually produced seeds from the same organism, right? Not possible with a lot of other organisms. So, what traits did Gregor Mendel keep track of? 7 of them is what he was looking at. So he measured seed color and seed shape, and he documented all this, right? That's one of the important things about being a science. You have to document things, and so he wrote these things down and kept data tables and kept the numbers and observations. So let's start off with making sure we're all on the same page. What is fertilization? Well, in this case, we're talking about sexual reproduction. Okay? And so fertilization, for our purposes right now, is a process by which male and female reproductive cells join together and they form a zygote, right? In peas, that is the zygote is the embryo, and your seed is sort of surrounding that. Okay, so you can sort of see the picture there. So, reminder again, I hope, types of cells in organisms, right? Remember, there's somatic cells. That's all of our other cells, right? Our brain and lung and nerve, and all of our body cells are considered somatic cells. We're talking right now about the sex cells, right? So these are otherwise known as gametes, right? And obviously in humans, we're talking about a sperm and an egg. In plants, we're talking about pollen and egg. Same idea though. When these sex cells combine, you get a gamete. I'm sorry, you get a zygote. These are gametes. When they combine, you get a zygote. Okay, so now from how does this stuff connect to where we're going with genetics? Okay, So to be fair, I want it to be understood, Gregor Mendel did not know what a gene was. He didn't even know much about cells, right? At that point, there weren't powerful enough microscopes, and it really wasn't understood until later in the 1800s that chromosomes even existed. And so Gregor Mendel referred to these things as factors. And what was really interesting is the data that he collected on these factors. Okay? We now know that the factors he was referring to are genes or sections of DNA that code for traits. Okay? So, we know that we don't all have the same traits. So how do we have different traits? Well, guess what? We have variations of our genes. And those variations are called alleles. We also, much like the pea plants, have two copies of any given gene. So it's a little bit redundant. You might ask yourself why, and hopefully you'll come to the conclusion why is because you got one from mom and you got one from dad, right? Dad contributed a version of what your hair should look like. Mom contributed a version of what your hair should look like, let's say, if those were Mendelian traits, which it probably isn't a Mendelian trait, but all right. And from that, you got your hair color. Right? Well, it's the same thing in peas, right? The female and the male portion, right? We're both contributing factors for what the flower color would look like, or what the seed color would look like, or what the seed shape would be, right? So those are alleles, variations of genes. Some more terminology, lots of terminology in genetics, is how do we, without just giving these things big long names, right? The section of DNA that codes for hair color, right? Instead of calling it that, we are just going to assign it a letter, right? So maybe a capital G or a lowercase g to tell us the difference between the alleles we're looking at. So when we put those together, that's called a genotype. So a genotype is simply the alleles that make up a trait, represented by letters, right? So this would be your genotype, capital G in this case, or homozygous dominant, which we'll come to in just a minute. Phenotype, I always remember, is the physical trait. What does the organism look like as a result of its genotype? So in this case, if capital G stands for green eyes, this particular fly looks like it got a copy from mom and a copy from dad that both said green eyes, so there wasn't really going to be any other option but the phenotype being green eyes in that fly. Okay, so let's review a little bit, remind ourselves what type of cell is this, and it's easy to say it's a sperm cell, but it's a gamete, right? What's the relationship between a gene and DNA? I like to think of these things as kind of like words and sentences and paragraphs, right? DNA are like letters, right? And we string together enough letters, we make words. Well, genes are kind of like the words, right? You string together a bunch of DNA and it starts to form words and sentences, right? If we put enough of them together, we get paragraphs, and enough together, we get chapters, and we get books, right? You can think of DNA being organized in much the same way, right? So DNA is the smallest, genes are in the middle, chromosomes are the next bigger, the full nucleus and genome would be like the whole library full of information. Okay, and then finally, would you say this is a genotype or phenotype? Hopefully you would know these are genotypes, right? Letters representing alleles. Okay, so what did Mendel do exactly, right, to generate the data that he came up with? Well, he ran 3 tests, essentially, over and over and over again. I think the numbers were crazy. He sampled and looked at thousands and thousands and thousands of peas over the years that he was doing this. And so his first tests were really, he was manipulating the flowers of the pea plants to be true breeding. So he made sure that the only pollen and egg connection of a white flower was from that same flower. So he was literally self-pollinating, right? Again, this can't be done in other eukaryotic organisms. Because plants can do this, he was able to then create true breeds or purebreds of white flowers. And he did the same thing with purple flowers, right? So he self-bred together all of his white flowers and all of his purple flowers, so that after a while all he got was plants that produced either purple flowers or white flowers. right? Those were his sort of true-bred plants, purebred plants. He then moved to cross-pollination, right? And cross-pollination is just like it sounds. Now he combined the pollen of one plant to the egg of another, or the stigma of another plant, right? So now he combined that purebred white flower with his purebred purple flower. And this is where the data started getting surprising. Instead of having a blend or some of each, he found that of the 7 traits he looked at, all of them had one trait that when the purebreds were put together was the dominant trait. So in the case of purple and white flowers, when he put those 2 purebreds together, he ended up with all purple flowers for the offspring. That surprised him a little bit. Finally, on his 3rd test, he went back to true breeding again. So now, let me just go back here a page. He took these purple flowers and he bred them back with each other again. Okay? And this is where it got surprising. Right? So he did true breeding again. And now, instead of being all purple flowers, even though all the offspring of that first generation were purple, when he bred those purple flowers together, which of course would be the equivalent of, brothers and sisters is sort of being bred together. You can do that in plants again, right? He found that some of them were white that came back in that second generation, right? We've all heard of traits hiding for a generation or whatever. Well, this is where that started, that idea came from. And he didn't just find that some of them, he found patterns in his numbers. And that was what was so interesting. He found that exactly 25%, or very close to 25%, of the flowers born in this generation were white. So to put it all together, it's a little confusing here, but this way you can see them all. Step 1, Step 2, and Step 3 of what Gregor Mendel did. Okay. That's enough for today. Hopefully you were able to get the notes down. If you want to go back through it, it's there for you. And we're going to move forward next time with Mendel's laws. So take care. I hope things are well.